So we're going to begin talking about microbial metabolism. Alright, so this lecture is split up into three major parts. We're going to talk about metabolic pathways and energy. We'll then talk about enzymes, and then we'll finish up by talking specifically about the three different types of metabolic pathways involving central carbon metabolism. Alright, so respiration and fermentation. Alright, so to begin with, let's talk about simplified model of metabolism. Alright, so whenever you're talking about metabolic pathways, alright, you're talking about two sides of the same coin. Alright, so you're talking about the nutrients that organisms are bringing in. Alright, so for instance here we have glucose, alright, that's entering a cell. Now this glucose molecule has potential chemical energy. Now this potential chemical energy has to be captured in some way, it has to be freed. Alright, so it goes through a process of what we call catabolism. Alright, so think about catabolism, think about breakdown. Alright, so you're basically taking glucose and breaking it down into its individual components. You're stripping it of its energy. And the energy you're stripping it of is in the form of the chemical bonds that hold that particular molecule together. So you're basically removing electrons. Okay, so eventually this captured energy is going to be converted into, at the very end of this process of glycolysis and Krebs and ultimately a respiratory chain, you get ATP. Now this ATP is what allows for the other series of reactions. Alright, so you have your anabolic reactions. Alright, so these are more of your building or your synthesis reactions. Alright, so here you basically have a cell that is going to use this energy and some other precursor molecules that is either assembled itself or brought into the cell, is going to use the energy of ATP to make much more larger molecules, alright, from basic building blocks. Alright, so amino acids, sugars, nucleotides, fatty acids. Alright, you're talking about your monomers, and you're basically building your polymers. Alright, so this should be review from the first chapter. Alright, so basic biochemical molecules, biological molecules of cells. So these much larger molecules and structures require the expenditure of ATP, and all this is ultimately used to add onto the cell and to create new cells. Alright, so when you talk about metabolism, right, you have a series of chemical reactions that don't go from basically A to, product F. Alright, that isn't the case. That would require too much energy. Alright, so you go from A to B, B to C, C to D, D to E, and so on, and each step in the process requires the use of a specific enzyme. Alright, and each enzyme is only going to target one of these reactions. Alright, so again, catabolic and anabolic pathways. Alright, so organisms take in food in their diet. This food gets broken down through catabolic pathways like cellular respiration, and you end up with basic building blocks. Alright, you end up with atoms that can be rearranged and used to build larger macromolecules. And in this process, you're getting the building blocks you need, but you're also capturing energy in the form of ATP. Alright, so you're taking the potential chemical energy stored in the food, you're releasing it by breaking chemical bonds, you're capturing that energy, and you're using it to make this intermediate, or this high energy intermediate ATP. Alright, the ATP is then used to drive your anabolic reactions. Alright, so taking these basic building blocks, like for instance amino acids, and making larger macromolecules like polypeptides or proteins. Alright, in this process, the ATP is going to be hydrolyzed. Alright, so you add water and get ADP and this inorganic phosphate. Now we'll talk about what this inorganic phosphate does. Alright, but the breaking of this bond releases energy. Alright, and that energy drives the synthesis of these larger molecules. Okay, so alright, thinking back to structure of an atom, and you recall that the electrons are orbiting outside the atom, alright, in different electron shells or orbitals. And at each shell, alright, as you get further and further away from the nucleus, you have more potential energy. Alright, now, why is this exactly the case? Well, think about it. This has a nice negative charge. This has a nice positive charge in terms of your nucleus. Alright, so the further away you are, the easier it is to kind of pluck off this electron or remove it. Alright, the closer you get to the nucleus, alright, the harder it is to pull away the electrons. Alright, so looking at potential energy and chemical energy. Alright, so potential energy, alright, being stored in biological molecules or molecules in nature. Alright, so here you have H2O and oxygen gas. Alright, and you're forming water. Now, in this case, you're going from something that is less organized and random. It has, in this case, a higher amount of potential energy. Alright, so you have your hydrogen molecules, you have your oxygen molecules, and then here you have your little water molecule. And you go from something that's less ordered to something more ordered. Alright, so in this case, the amount of chemical energy in this case, you actually lose potential energy. Alright, because these chemical bonds and the hydrogen or molecule, hydrogen gas and oxygen gas molecules gets broken, release potential energy, and the new bonds that form, there's less potential energy in those molecules. Alright, so this kind of ties back to the idea of first and second laws of thermodynamics. Alright, so in terms of energy, conservation of matter, conservation of energy, that energy cannot be created nor destroyed. Alright, so basically what you start with is what you get in the end. Alright, so here you got a little bear eating fish. Alright, that fish has stored potential energy. Alright, as that bear is digesting that fish, the energy stored in that fish is going to be released, and the bear is going to capture it through cellular respiration. Alright, the second law of thermodynamics has to do with the fact that entropy is increasing. Alright, so the state of disorder. Alright, so always going towards a state of disorder. Alright, what this also means is that your metabolic processes are not perfect. Alright, because in this case, the bear breaking down the fish and capturing the energy stored in that fish, he isn't going to capture 100% of the energy. Alright, a lot of that energy that's stored in that fish is actually going to be released in the form of heat through this process of cell respiration. Alright, he's also releasing CO2 gas and water, but he's also going to be making ATP. Alright, but this is not a 100% efficient process, right, because of the second law of thermodynamics. Alright, so thinking about entropy. Alright, so entropy has to do with the movement of molecules. Alright, so going from order to disorder. Alright, so looking at a solid, this is a very ordered structure. Alright, so thinking about solid water, highly ordered structure. You have a nice little crystallized structure. Liquid water, alright, the water molecules are still interacting with one another through hydrogen bonding. They're still close together, but not as close as solid. Alright. And so, to kind of give you an example of this process of going from something highly ordered to something less ordered. You're going from ice cube that's basically melting, all right, over time into a puddle of water, or something that's highly structured to less structured. All right, the water molecules spread out, they're no longer organized, and then the highest level of entropy in this case would be gas. All right, so here, this kind of ties into food chains. All right, so thinking about a food chain, all right, so you have your primary producers, all right, so your photoautotrophs, all right, that are using the energy from the sun, the light energy, they capture to make food. All right, now you have 10 joules of energy that's available as food in this little plant here. And so now you have some herbivore that comes along. So in this case, you have a chemo heterotroph comes along, feeds on this plant, all right, and it gets 10 joules of energy from the plant, but basically not 190 joules of the energy is actually lost to the environment. All right, so now this little herbivore now has 1 joule of energy available, all right, stored in it, and you have another chemoheterotroph comes along and feeds on this little deer antelope, and it captures the energy from it. Again, 9 joules of energy is lost to the environment. All right, it only captures a small percentage of the energy stored, so like 1% of the energy stored. And so on. All right, so basically this here, this loss of energy to the environment is due to entropy. Alright, so thinking about this idea of entropy. Alright, so think about which is more likely. If you're driving down the road and you're sitting in the back of a truck and you're tossing bricks out, are they going to be in a nice little stacked pyramid, or are they going to be disordered all in a jumble? Well, this is the more probable scenario. Alright, so that doesn't take a lot of work. Alright, but it does take work to take something that's disordered, like a pile of rubble, and to make it ordered. Alright, so this takes work. In this case, work involves energy, involves energy, or ATP. Alright, so talking about energy. Now, when you start talking about chemical reactions that takes place in a cell, alright, you're talking about free energy. Alright, so this free energy is what we call Gibbs free energy. Alright, so Gibbs free energy is the energy that is able to do work. Alright, so delta H, as I mentioned, is basically enthalpy. It measures the chemical potential energy. Alright, and then delta S measures the amount of disorder. In this case, temperature is in degrees Kelvin. Alright, so we're going to eventually just focus on Gibbs free energy. Alright, that's the big picture. This is Gibbs free energy, because this is what the cell is actually using to do work. Alright, so when you look at a chemical reaction, and that chemical reaction, you can be shown the delta. Alright, so this delta is referring to a difference. Alright, so I'm talking about final minus initial. Okay. So if delta G is less than 0. All right, you have a spontaneous reaction. All right, so spontaneous chemical reactions occur on their own. All right, the cell does not have to invest any extra energy into the process. All right, so another name for a spontaneous chemical reaction is a exergonic reaction. All right, now these reactions also release energy. Okay. Now, if delta G is greater than 0, the reaction requires energy input. Now, this energy input is in the form of ATP. All right, so to get these chemical reactions going, you have to invest energy. All right, so we'll talk a little bit later about thermodynamic barriers to chemical reactions. All right, but here, a non-spontaneous reaction is also referred to as being endergonic. So these reactions are going to be absorbing energy. Okay. Now, when delta G is 0, the reaction is at equilibrium. Now, if a cell is at equilibrium with its environment, it's a dead cell. Okay. Alright, so looking at a couple examples involving free energy. Alright, so you have this diver on top of a diving board. Alright, so there's a high amount of potential energy here. Alright, now when a diver jumps off, alright, that energy is released, and when he hits the water, alright, there's the lowest amount of free energy. Alright, so you go from high energy to low. So you go from basically being less stable to more stable. Alright, so here delta G becomes negative. Alright, so here you also have, for instance, little bitty molecules, particles, and they're bumping into one another, and they're going to, as they're bumping into one another, they're going to diffuse. So you're going from something ordered to something less ordered. Alright, so low entropy to higher entropy. Alright, so here it's like taking a molecule of glucose, for instance, and breaking it into CO2 and water. Alright, so you have a highly ordered structure. Alright, so again, low entropy, and then going into releasing the chemical energy stored in this molecule, and you end up with water and CO2 gas. Alright, this is less ordered, higher entropy. In all these cases, it's describing a spontaneous process that's going to be exergonic. Now, looking at these exergonic reactions. Alright, so here you're looking at the amount of free energy. Alright, so this is just a generalized reaction. Alright, so your reactants have a higher amount of potential energy than your products. Alright, so as a result, you're actually going to be releasing energy in the process through this chemical reaction. You're releasing energy, and here, the overall difference, alright, so your product value, alright, is going to have less energy. Alright, so the difference in this gives you a negative delta G value. Alright, so it's less than 0, so as a result, this is a spontaneous reaction. Now your inorganic reactions require energy. Notice here your reactants have a lower amount of free energy than your products. Alright, so in this case, instead of releasing, you're actually going to be investing energy into the reaction, typically form of ATP. Alright, and you're going to take your reactants and form products. Alright, so you're going from, in this case, less ordered, like let's say taking some amino acids and making a polypeptide. Something that's more ordered, alright, that has more potential energy. It's a bigger molecule, alright, so it's kind of also taking, like, thinking about taking glucose, alright, a monosaccharide, and making, for instance, something like starch, or cellulose, or glycogen. Alright, all these molecules have a large amount of potential energy. Alright, so the best way to think about exergonic and inorganic reactions. Alright, so exergonic reactions are spontaneous. They occur on their own. Alright, so it's kind of like having a ball at the top of a hill. Now, as you release the ball rolls down the hill. Alright, and as the ball is rolling down the hill, that energy is released. Alright, in this case, energy is released, and it's allowing for this ball to move. Okay. Now, in an inorganic process, you have a ball at the bottom of the hill, and you got to push it up to the top. Now, this requires energy. Alright, the ball isn't just going to spontaneously roll up to the top of the hill all by itself. You have to invest energy. Alright, so in this case, chemical reaction would take ATP. In this case, you'd physically have to push the ball up the hill. Alright, so we're talking about these two types of reactions separately, but in reality, they're actually interconnected. Alright, so your exergonic reactions. Alright, you'd have your high energy reactants, something like your glucose, and here you have your byproducts of respiration, CO2 and water, alright, and you're producing ATP. Alright, so that's the exergonic process. And then here you have an endergonic process. Alright, so this ATP is used for something. Alright, so here you have low energy reactants, let's say something like an amino acid, and you're going to make a high energy product like a protein. Okay, but these are coupled processes. One, your exergonic reactions release energy, your endergonic reactions use that energy to drive biological processes, reactions that need energy. Alright, so another important topic to discuss is oxidation and reduction. Alright, so whenever you're talking about oxidation reduction, the way that I remember these two terms as they're applied to chemical reactions. Alright, so there's a cute little acronym. Alright, so Leo the lion says grr. Alright, so whenever you're talking about oxidation and reduction, alright, so if a molecule is oxidized, what it has done is it has lost electrons. if a molecule is reduced, it has gained electrons. Alright, so here you have a kind of arbitrary fictitious molecule. You have molecule A and molecule B. Alright, so these are our reactants. Alright, so molecule A has electrons, molecule B doesn't. In this case, it's the one that's going to be receiving the electrons that it needs. Okay. Alright, so oxidation and reduction reactions are nothing more than shuttling around electrons from one molecule to another. Alright, so it's a dance. Okay, so molecule A will transfer electrons to molecule B. And your products, alright, molecule A has lost electrons because it has donated the electrons to B. So A becomes oxidized, and B has picked up these electrons. Since it gained electrons, it's reduced. Alright, so loss of electrons, oxidation, gain of electrons, reduction. Now, how does oxidation and reduction tie into what we're going to talk about in terms of metabolic pathways? Alright, well first, let's talk about a basic example of oxidation reduction. Alright, so sodium and chlorine. Alright, so sodium, alright, to become an ion, a cation, it gives up one electron. Alright, so your sodium ion is going to lose one electron, and your chlorine atom is going to gain one electron. And so in this process, your sodium atom actually is going to, since it has lost an electron, it becomes oxidized. And since the chlorine atom has accepted an electron, it's gained an electron, it is reduced. Alright, so later on we're going to start discussing, when we talk about cell respiration, we'll talk about these carrier molecules. Alright, so these carrier molecules are called electron carriers. Alright, so electron carriers, hydrogen atom carriers. Alright, so you have two specific molecules. You have FAD and you have NAD+. Alright, so both these molecules function as electron carriers in cellular respiration. Alright, so what happens here is your FAD molecule basically acts like your little electron carriers are basically a taxi, and they shuttle around electrons. That's their job is. They transfer electrons from point A to point B. Alright, they're taxi cabs. Alright, so FAD picks up hydrogen atoms. Alright, so think about it. A hydrogen atom has one proton, one electron. Alright, so you can think of it as an electron in this case. Alright, so FAD can pick up electrons. Alright, so FAD picks up electrons. Alright, so it gains electrons, so it's going to be reduced. So it becomes FADH, two hydrogens, 2. Alright, so FADH2 is reduced. Alright, so now FADH2, it can donate or give up its electrons, and in the process of donating and giving up its electrons, alright, it's going to be oxidized back to FAD. Now, the other electron carrier molecule is going to be NAD+. NAD+ can pick up its hydrogen atoms. Alright, so this becomes NADH. Okay. NADH is reduced. all right, NADH can give up its electrons, it's oxidized back to NAD+. Alright, so here, let's look at another example of oxidation reduction involving a chemical reaction. Alright, so you have methane, you have oxygen gas, alright. So this is a combustion reaction. Alright, you produce CO2, energy, and water. Alright, so look at methane. Alright, so methane is a what? This is what we call a hydrocarbon. Alright, so notice that in methane, the electrons are located centrally. Alright, so this is actually a non-polar, alright, covalent bond. Alright, so your non-polar covalent bonds, this is a non-polar molecule, alright, and it has a high amount of potential energy, alright, stored. Alright, so you can break these chemical bonds and release energy. Now what happens is actually this energy is released. It's given up to and used to make ATP. Now, as you're breaking these bonds, you're releasing electrons. Well, where do those electrons go? Well, those electrons are eventually donated to oxygen, okay, to form water, alright, because oxygen picks up electrons, in this case hydrogen atoms, and is reduced to water. Alright, so look here, you have methane with 4 hydrogens. Alright, now those 4 hydrogens get broken off, and those 4 hydrogens are used to make 2 molecules of water. Alright, so again, tying in this idea of conservation of energy, conservation of matter, what you start with is what you end with. Alright, so here you're being shown a picture of NAD plus NADH. Alright, so NAD plus, right, notice here that you have a nice little phosphate molecule, All right, you have a ribose sugar, and you have nicotinamide. All right, so this would be an example of a nucleic acid. All right, so here, NAD plus, you have that little positive charge there in the nitrogen. You accept your two hydrogens, and they go here, and you end up with NADH plus a proton. Alright, so looking at FAD and NAD plus. Alright, so here you have some arbitrary reactant. This arbitrary reactant is going to give up its energy, potential energy, to FAD. Alright, so in this case, this molecule is going to be oxidized, and this molecule will be reduced. Alright, so A, reactant A with two hydrogen atoms, gives up electrons, gives up its hydrogens to FAD. Alright. Alright, so this initial reactant is going to be oxidized, it's lost electrons, and FAD accepts these electrons and is reduced. Alright, so electrons are being shuttled from one molecule to this molecule here, an electron carrier. Alright, so the same thing here. Alright, so with nicotinamide, a adenine dinucleotide. Alright, so you have some arbitrary reactant with two hydrogen atoms. Alright, you have NAD plus. Alright, so this molecule here will be oxidized. This molecule here gets reduced. Alright, so reactant B gives up its hydrogen atoms, its electrons, to NAD plus. In the process, reactant B is oxidized, and NAD plus is reduced to NADH. Alright, so now NADH can shuttle electrons to somewhere else in the cell. Alright, so here, alright, you're being shown now a cyclical process, alright, where you have here NAD plus, and you have an electron donor. Well, both of these reactants bind to their active site, their binding site, in an enzyme. Alright, here you have the exchange of electrons from your electron donor to NAD plus. Alright, so your electron donor, it gives up electrons, it becomes oxidized. NAD plus accepts the electrons, it becomes NADH. Now NADH will transfer those electrons to some other molecule. Alright, so here NADH comes in and binds to another enzyme. You have another molecule that's an electron acceptor molecule, or a molecule that can accept electrons. Alright, and here you have the electrons that are transferred from NADH to this electron acceptor molecule. Alright, in the process, NADH becomes oxidized to NAD plus, and your electron acceptor molecule is going to be reduced. Alright, so again, coming back to this idea of ATP. Alright, so ATP is basically the currency of the cell. Alright, that's what gets exchanged. Alright, that's what it takes to do work or to perform some job. Alright, so you should recall that ATP, you have adenosine triphosphate. Alright, so here, alright, you have 3 phosphate groups. Now these 3 phosphate groups don't like to be near one another because they have negative charges. Alright, and negative charges close to one another repel. Alright, so that puts a strain on these chemical bonds that hold the atoms together, hold the molecules together. Alright, so basically they're so unstable that basically when you break this bond, you release a tremendous amount of energy that can be used to do work for the cell. Alright, so here you have ATP, you add water, alright, so through a hydrolysis reaction, alright, you break apart ATP into ADP and inorganic phosphate, and you also release energy. Now, this energy can be used to do work. Alright, so again, remember, your exergonic reactions are coupled with your endergonic reactions. Alright, so here, on the y-axis, you're shown the free energy relative to this. Alright, so your endergonic reaction, A plus B. Alright, so here, there is a low amount of free energy. Alright, and you're trying to make a molecule that has a higher amount of free energy, AB. Okay, so you're trying to synthesize a molecule, a larger molecule. Alright, so this is a endergonic process. It requires energy. Alright, so you take an exergonic process where you actually produce and release energy in the form of ATP. Right, so energy gets released, and in this process, energy being released gets captured and transferred to these original reactants. Alright, so the phosphate group in ATP that's produced from your exergonic reaction gets transferred to reactant B. Alright, so reactant B has a higher amount of energy. Alright, so now it can react with reactant A and you form your product, AB. Alright, notice that the energy released when you synthesize BP is going to be the same energy that's released to synthesize AB. Right? They're equal. Alright, so here you're looking at chemical work. Cell can use ATP for chemical reactions. Alright, so you have glutamic acid, alright, plus ammonia gives you glutamine. Alright, so notice that this has a positive delta G. Alright, so this is a endergonic process. Alright, you're going from something with a low amount of potential energy to higher potential energy. You're making a larger molecule, so this has a positive delta G value. Alright, now you can take ATP, you can hydrolyze it, the phosphate group gets transferred to the glutamic acid. Glutamic acid then has a higher amount of potential energy. It can interact with the ammonia and form glutamine. Alright, so what's going on here? Alright, now this reaction takes by itself to go from glutamic acid and ammonia to form glutamine, right? A positive delta G value, plus 3.4 kilocalories per mole. All right, you add in ATP, the energy that's released from taking the ATP and hydrolyzing it into ADP plus the phosphate, which that phosphate gets attached to the glutamic acid initially, and then it's broken off and you form your glutamine. But the initial hydrolysis of this phosphate, energy is released in the form of negative 7.3 kilocalories. Alright, well, if you take 3.4 plus negative 7.3 kilocalories, you get a negative delta G value. So now this reaction has a negative delta G value. So this is a exergonic process. So you take a intergonic process and make it exergonic. You make it so that this reaction can actually take place. Alright, so your catabolic reactions produce or allow for the production of ATP. Your anabolic reactions use ATP to make larger molecules. In the process, you break apart your ATP molecule into ADP and inorganic phosphates, which then your catabolic reactions use the energy from molecules like glucose, alright, to assemble ADP and phosphate back into ATP. Alright, so it's one big cyclical process. Alright, so here's kind of a concept check question. In a redox reaction, loss of electrons is what? Is it phosphorylation? Is it oxidation? Fermentation? Reduction? Or none of the above? Alright, well, if you're losing electrons, it's oxidation. So remember, loss of electrons, oxidation. Gain of electrons, reduction.